Okay, hello everyone, my name is Gary Lee Clemens. I'm a student here at the University of New England Pre-Health Program, and today we're going to be going over, or rather we're going to be exploring three very different cellular pathways, okay? The route for a lipid steroid hormone, the route for a protein that's exported from the cell, and then a route for a protein that stays and works inside of the cell. All right, our three examples will be cortisol, growth hormone, and actin. Each one begins at a different starting point in the cell and uses a different set of organelles and ends up in a very different place. All right, so by following each pathway step by step, we'll see how the cell organizes itself to make sure that every macromolecule reaches the right destination. Okay, so the first one is going to be, we're going to talk about the cortisol steroid hormone, right? In which we have the cortisol pathway. Now, cortisol begins as cholesterol, which the cell obtains from its own metabolism or from the bloodstream. Cholesterol is then found in a cytosol and serves as the raw material for many steroid hormones. Okay. Next, let's talk about the smooth ER, in which the conversion of the cholesterol into cortisol takes place in the smooth endoplasmic reticulum. This organelle contains specialized enzymes that carry out a series of chemical reactions, right, step by step, in order to produce the cortisol. Next is the transport vesicle. So once the cortisol is made, it can't just float anywhere. It has to get packaged into small transport vesicles that carry it onto the next stage. Next is the Golgi apparatus, in which at the Golgi apparatus the cortisol is further processed, sometimes combined with the carrier protein, and then stored into the correct pathway for export. All right, so moving on, from here, it's placed into the secretory vesicles that move toward the cell membrane. Okay, and then finally, because the cortisol is then lipid soluble, it diffuses right through the cell membrane into the bloodstream, where it can travel to its target tissues throughout the body. Okay, moving on to pathway 2. So for pathway 2, we're going to discuss the growth hormone, right? The protein for export. Growth hormone story begins in a very different place, the rough endoplasmic reticulum. Here, ribosomes attached to the endoplasmic reticulum, the ER surface, build the amino acid chain that will become growth hormone. As it's made, the chain enters into the ER lumen, where it folds into a basic shape and then begins to receive those chemical modifications. Okay. Next, we'll talk about the transport molecules, in which the protein is then loaded inside of those vesicles that carry it away to the ER. At the Golgi apparatus, the growth hormone undergoes further modifications, such as glycosylation, and then is carefully sorted for its role as a secreted protein. And then the finished hormone is stored in these secretory vesicles, near the cell membrane. Now, these vesicles don't release their contents right away. They wait for a signal, such as a chemical messenger, and that tells the cell, hey, it's time to secrete that particular growth hormone. All right, and then once the when the cell arrives, the vesicle fuses with the membrane, releases the growth hormone into an extracellular space, and then eventually, obviously, into the bloodstream. And this process is known as, in the very end here, is known as the exocytosis. Next, let's move on to pathway number 3. Okay, so here we're going to be talking about actin, right? An intracellular protein. Actin is made by free ribosomes floating in the cytosol. And because it's not destined to leave the cell, it's one of the most common proteins in the cell. Okay. After translation, actin monomers quickly assemble into long, thin filaments, and this assembly is tightly controlled so that the actin can be built or broken down as needed. Now, these filaments join the cytoskeleton the cell's internal framework. And here, actin helps maintain the cell's shape and enables the movement and even can assist in moving the organelles inside the cell. So in closing, by comparing these three examples, we can see that the cell uses very specialized pathways, depending on the type of macromolecule. Cortisol relies on the smooth endoplasmic reticulum and can diffuse outside of the cell. Growth hormone is built on the rough ER and is processed in the Golgi apparatus and then released through exocytosis. And actin is made in the cytosol, but it never leaves, becoming part of the cytoskeleton. This coordination ensures that every molecule is in the right place at the right time, whether that means being sent across the body or staying at home to keep the cell structure intact. Here are two of my references that I utilize. Feel free to contact me if you have any questions.